Well, Jim Groombridge is Reader in Biodiversity Conservation at the University of Kent. His research interests are many, but the central focus of his work is on the theoretical and practical aspects of endangered species conservation, and in particular, the application of studies to enhance our understanding of the biological processes. Today, he's going to tell us about work carried out on the critically endangered Seychelles paradise flycatcher. Please welcome Jim Groombridge.
Thank you very much. I should first say thank you very much for the invitation to speak. It's a pleasure to be here and also to see some familiar faces in the audience, including some members of the trustees of the African Bird Club, including some who are members of the MSc program at DICE at the Durrell Institute of Conservation and Ecology, which is based at the University of Kent, where I currently work. So I'm very pleased to be here. Thank you very much. So today's talk is about the Seychelles paradise flycatcher. I want to give you an update on some of the work which began back in 2007 to try and restore this critically endangered bird species. This was a complicated project. It was funded by the Darwin Initiative, so UK government DEFRA funding, but it involved a large number of organisations, which I'll acknowledge in a few slides' time. So just to give a brief background, so I lead a conservation genetics research group at DICE with a collection of people that do applied conservation genetic work. But when I say applied, we work very closely with partners all over the world in various places who work with managing critically endangered species, and we work with them on reintroduction projects, and other types of practical conservation initiatives where often genetic data as well as ecological data is needed to enhance those conservation initiatives. And I should point out the work that I'm going to talk about today is largely been done by Rachel Bristol, who's now Dr. Rachel Bristol. She completed her PhD at DICE a few years ago. So we have a genetics lab, and there's a few forced smiles there for the camera, but we're all enthused about applying genetics and science to try and enhance the success rates of conservation work. So on a broader perspective, reintroductions, which is what I want to focus on, these are quite an expensive and logistically difficult occupation for conservation biologists. And if you look back in the literature, in the 90s, people who looked at sort of global levels of success saw about only 1 in 10 reintroductions where it could actually be deemed successful. So there's actually still a long way to go to actually try and make this type of occupation, this type of initiative more successful. So from a genetic point of view, most species that are endangered or critically endangered, there'll be a period of population decline and followed hopefully not by extinction but by some level of recovery as a consequence of conservation effort. And one of the things that we're very conscious of is that in reintroductions, where a subset of individuals are moved to another place to establish a new population, that there's quite often what we would call a secondary bottleneck effect, where we would be there's a risk of the loss of genetic diversity in the reintroduced population as a consequence of the individuals that have been selected. So this is a sort of a broader perspective to take on the rest of this talk, and I'll come back to this point in the last few slides. So alongside all of that and reintroduction and all the efforts that go into it, the extinction vortex is what we use as a catch-all phrase to describe the population decline and demise of endangered species, and this includes habitat loss, introduced invasive species, emerging infectious diseases, and all the other sort of culprits of the loss of biodiversity that we fight against as conservation biologists. And the Seychelles paradise flycatcher is a remarkable species. It's one of the last critically endangered species in the Seychelles. It's endemic. And it's a striking bird. It's very easy to see if you go to the Seychelles, it's you know, as soon as you get off the ferry on La Digue Island, where it's found, it's one of the first species that you would see. So it's quite in that respect, it's quite a straightforward species to work with. And I should acknowledge these partners. So all of this conservation work, in particular the translocation, was undertaken, funded by the Darwin Initiative, but Nature Seychelles were the main host country partner. We also worked very closely with the government of the Seychelles, but also with Dennis Island, which is the island where we moved the birds to. The Wildlife Vets International, which is the veterinary organisation that we work with very closely on all of the projects throughout the world that are part of this group, for disease screening and the health of the birds. And RSPB2, and it's good to see a few familiar faces here, including Paul Buckley, who was involved in much of this work in its initiation. So it's the last critically endangered species in the Seychelles. This is the picture of the male. They have these remarkably long, striking tails, and they are thought until 2007 to be a population size globally of less than 250 individuals. All of them found on the island of La Digue in the Seychelles. So La Digue is here, so a tiny island. It's there aren't many cars on it, and it's a place that most people travel around on foot or by bicycle. You could cycle around it in a day to give you an idea of the size of the place. Again, this species is found only there. Historically, it was found probably across most of the major islands in the Seychelles, and historical records sort of confirm that. This is an aerial view of La Digue, and it shows that the most of the human population of around about a local population of about 2,000 people live on the west side, which is the sort of the lowland plateau at around between 3 and 5 meters altitude, and then there's a sort of what they call the hill the hill on La Digue, which is less populated by people. But flycatchers actually cover most of the island, and in actual fact, they are commonly found in people's gardens in the Seychelles on La Digue. So they are, a very easily accessible species to see. One of the pressures for the species, the threats, has been the tourism industry. Clearly, with small islands, the pressure on land is such that there has been habitat loss, and that has caused the decline and the local extinction of this species from some of the other outer-lying islands in the Seychelles. And it is still continuing, but we're hoping that it has slowed as a consequence of some of the work that has been done on this species. So it's a critically endangered. It's restricted to a single island, or it was until we began this initiative. And largely due to habitat loss, there will have been other factors, invasive species such as rats and cats, and other competition from other introduced bird species. And the plan has always been to, the species action plan has been to restore this species to 3 or 4 additional islands to create safety net populations. And if you look at the evidence for its the sort of chronology of its distribution, it's really marked by this sort of local extinction across various of the outer-lying islands, including Praslin, where it was seen until 1989, but it's now no longer found on that island. And even on La Digue, the most extreme part of this species population bottleneck was in the 1960s, it's believed, as a consequence of surveys that were done at that time. But actually, we've actually seen something of a recovery. If this is if this trend is actually a reflection of the population's true trend, then it has been something of a recovery itself over the last 30 or 40 years. But nonetheless, the population on La Digue today has been is still very, very small. So as a consequence of this period of decline and the status of the species, we began to think about reintroducing the population to an additional island. And the choice that was made was Dennis Island. So Dennis Island is up in the north here. There are a lot of islands to choose from. They all have particular characteristics. Dennis was chosen. We did a lot of work to survey all of the islands for their suitability. The flycatcher is a species that nests in broadleaf forest, takamaka trees, and there's a good habitat for that on Dennis. It's also rat-free because it's had rats eradicated from it several years ago. So it had all of the hallmarks of a good place to put flycatchers as a first reintroduction effort. And perhaps the first thing to do was to ask, well, if we're going to work on this species, how evolutionarily distinct is it from all of the other flycatcher species that are found across the Indian Ocean islands? And these are some museum skins of different flycatcher species and subspecies from different parts of the Indian Ocean. And it puts it into perspective nicely, the morphology of the male flycatchers on the Seychelles compared to some of the other islands. So this doesn't give you a true representation of the taxonomy because there are lots of different subspecies across, certainly in the Comores. There are 2 subspecies on Madagascar. There are 4 taxonomic subspecies in the Comores. And lastly, the mascarene flycatcher, which has taxonomically 2 subspecies, one on Mauritius and one on Réunion. The one on Réunion is reasonably common. The subspecies on Mauritius is threatened with population of estimates at around 100 to 200 pairs. So we did some analysis using museum samples where we extracted DNA from lots of the taxonomic distribution of the different flycatcher species. And this is Rachel, who based a lot of her PhD work on this study. And we created a phylogenetic tree. So this is based on over 4,000 bases of DNA, very comprehensive. And what it shows is a few key attributes. So what we were interested in was the evolutionary distinctiveness of the Seychelles flycatcher. And what's important here is the long internal branch length here that shows that the Seychelles paradise flycatcher has been isolated on Seychelles for a considerable period of time, but that actually the Indian Ocean islands have been characterized by perhaps up to 4 or even 5 separate radiations of flycatchers across the Indian Ocean islands. Something else to note is that the Réunion and Mauritius, essentially the mascarene flycatcher, which is found on these 2 islands, appears to be the most ancestral of the, essentially appears to be the first radiation into the Indian Ocean islands. So what this has helped us to do was to put in, shine a spotlight on the Seychelles flycatcher as worthy of conservation effort from a biodiversity and a phylogenetic point of view. So we began with a multi-partner project aiming to reintroduce birds from the source on La Digue to Dennis Island. Again, lots of partners involved. RSPB Nature Seychelles were the lead partners. We had Durrell on Jersey involved too. So lots of people to thank for this work. And the first thing we set about doing was to work with the local community to raise awareness of this species. So this is a species that's very visible, very easy to see if you're on La Digue, and so there's no reason why the local people didn't really feel that this was a particularly threatened species. So it was important to engage them in what we were doing and to dispel a few myths about the species, but also we learnt a lot from that process too, in learning about the biology and the ecology of the species. I also work with the school kids on our dig. Very convenient. There's a single school, so you get a good capture by working with the school kids. And again, you can raise awareness with the parents subsequently as well. So that was a really fun part of this Darwin project. And finally, it took us a year to really lay the groundwork for doing the translocation, which we undertook in November 2008. And this is our mist netting team on their local transport off to the mist netting sites. Andrew Greenwood there in the centre, he's the Wildlife Vets International avian veterinary specialist that we had on the team. And this is the environment that we were mist netting in, using playbacks fairly easy to catch these birds using mist nets and playbacks. They're highly territorial, so they respond to playbacks quite effectively. And also the working with the local people to help us along, so they were very good at working to mist net the birds, and also it's a very good educational tool and raising awareness about the threats associated with this species. And so we, in 2 days, we caught up, we translocated 23 birds. In fact, we caught up 35 birds. We actually had to release a few. We caught too many because the males were too enthusiastic in flying into the nets. So we had to throw a few back. But 23, so that represents about 10% of the world's population, to give you some idea of the plight of this species. There we are taking blood samples and putting colour rings on the birds that we were going to translocate. And so we had a mist netting team, it was an amazing experience to actually trap in 2 days that number of individuals. We kept them overnight in these relatively low-tech storage containers, donated kindly by the local supermarket, we put them in the back of a local taxi, took them to the helicopter pad the next morning, and flew them straight over to Dennis Island. So Dennis Island is a remarkable place to visit. It has a single hotel on it, so it's a sort of a 5-star hotel destination. But they have all sorts of other endangered bird species that have been reintroduced there, the hotel managers and owners take great pride in their looking after their reintroduced populations of birds. So we were met by the hotel managers and got on with taking the flycatchers out into the middle of the forest and releasing them. And it was a hard release, so it was we weren't putting them in aviaries and looking after them, and for a few days they were let straight out of the box as soon as they arrived. And there we are taking some, we gave them some sugar water before their release, and you can see the slightly worried look on my face, this is because when the helicopter departed with the first load of flycatchers, and we sort of waved it goodbye, it came over the radio from the conservation officer on Dennis that actually the night before there had been an arrival of between 30 and 50 red-footed falcons that had turned up. This is something that happens most years, and normally they get 2 or 3 individuals, and it's quite a rare event. And this year was a bumper year, so we had. So we were sort of waving with one hand, waving goodbye as a helicopter full of very expensively translocated flycatchers, and sort of having to cross our fingers really that these red-footed, how things were going to turn out, we weren't too sure. Anyway, we simply went ahead with it. Again, this shot shows you the remarkable turquoise colour of the eyes there. So these red-footed falcons were thankfully exhausted, and you could virtually pick them up off the floor. They were there just feeding on their way from the north to the south in their migration, and thankfully we had no problems with these, but it was a very nervous few days, I can tell you. So some milestones. We translocated 23 birds, 10% of the world's population. First breeding behaviour in January 2009. We actually moved the birds. We timed the translocation to be just before their main breeding season, so we wanted the birds to be full of hormones when they landed so they could kind of hit the ground running, if you like. Our first chick fledged in March in 2009, 5 successfully fledged offspring shortly afterwards, and then we were seeing second generation offspring breeding on Dennis Island not long after that. And here's some photographs. So all signs were looking pretty good, to be honest. And today, July 2013, this is a map from the most from a recent survey showing some healthy distribution of territories on Dennis Island, and the population has grown substantially. So this gives you an idea of the growth of the population, tripled in size in the last 7 years. You might notice the rather slow performance at the beginning here, and there's a reason for that, which hopefully has been fixed, and that is that introduced minors were on Dennis Island when we put the flycatchers on there, and during the sort of the early years of the monitoring of the population, we noticed that there was a lot of nest predation and disruption, and we didn't know which of the bird species, or whether it was introduced mammals on the island. But so we got some remote cameras, set them up on some nests, and identified them as common miners that were disrupting the nests. So fortunately, some other organisations began doing eradication program of common miners which are an invasive species on an introduced bird species on Dennis Island, we think this is been largely responsible for the upturn in the growth rate of the flycatcher population, so we're very grateful for that. Alongside all of that work, we also wanted to know, alongside the 23 birds, how many genes, what proportion of genes we'd carried from the source population on La Digue to Dennis Island. So Rachel Bristol did a lot of genetic work to look at this. So we developed some genetic markers. We took blood samples from all of the source population on La Digue, and also of the 23 birds that we moved. And then we were able to actually produce some analysis from which we could estimate for any given number of birds that we might translocate what proportion of genetic diversity we would move if we moved that number of birds. And you can see that actually, even if you only moved 5 birds, you'd expect to be moving over 70% of in this case, the neutral genetic variation in the population. So we were, on average, doing pretty well with our 23 birds here. That gives you an idea of what you'd expect, and that red dot is what we actually got. So that's in this case, the proportion of alleles in the population that we'd moved. So we actually, by chance, did slightly better than we might expect on average from this analysis. So one of the reasons for moving as much genetic diversity as you can with a reintroduced population is because you want to equip them with as much of the adaptive potential and genetic diversity for their future ability to evolve as a population in the long term. So that's one of the things we wanted to do, was to make sure that we were equipping this population as best as we could with the maximum amount of diversity so that it could evolve to future challenges. But we also wanted to look back in time. So Rachel took DNA samples from Seychelles Paradise flycatchers from the last 60 up to 130 years from historical museum specimens to look at levels of genetic diversity over a century ago and actually compare that to the amount of diversity we're seeing today. And this gives you an impression of what we found. So this is time along the bottom axis, and we see that there's been a dramatic reduction in levels of genetic diversity, in this case the mean number of alleles, in birds that were found that have been collected from the Seychelles in the 1800s, currently sitting in museum collections, through to the populations today. So around about almost a 60% historical reduction in genetic diversity as a consequence of the rarity of the species and the population bottleneck. If we look at if we go forward in time, we see that because we only moved 23 individuals, we missed 9.5% of the diversity that was there. So there are some alleles that we failed to move to Dennis with our 23 birds. But more importantly, the F1 generation, so this is the first generation of offspring on Dennis Island, it was an additional loss of genetic diversity. So what you see with every generation in any small population is a subsequent loss of genetic diversity as a consequence of genetic drift. And so this is what we were seeing today. So what we wanted to do was to put that loss of diversity through time within a reintroduction, but put it within a historical perspective over the last century. So these are some numbers that show you for different metrics of genetic diversity, how much was lost during the historical bottleneck over the last century, and how much in comparison was lost during the reintroduced bottleneck or the reintroduction. You might say, well, yes, there's large amounts of loss here and actually not a great deal of loss here. But actually, if you calculate it per generation, in which you can then compare the two, we actually find that the amounts of genetic diversity which were lost per generation during the reintroduction were actually far higher than they were during the rates of loss of the historical bottleneck. And so what this tells us is that, first of all, historical losses are fairly substantial, but if we compare that to rates of loss during the reintroduction, it's a lot bigger. So the upshot of this is that we need to be a lot more careful with how we manage reintroductions, and that trying to use genetic data to actually enhance and support reintroduction programs is actually very important. We can say quite a lot about it. And what we would hope potentially in the future is that if we move additional birds from La Digue to Dennis Island, then we would try and sort of move those birds that have alleles which aren't yet on Dennis Island. That work's not planned yet, but that in theory is what we would look to do. So looking outside sort of those two islands, elsewhere on the Seychelles, well, where to next? So the species action plan says that it's desirable to have flycatchers on 2 to 3 or 3 to 4 different islands. So where else could we put them? And there's potential in other islands. Curieux is a possibility. So this is an island which has some of the traits and characteristics that might be positive for flycatchers. And so there's some thinking going on at the moment about how we might be able to move towards that. But also more broadly, if you think back to the phylogeny, the molecular phylogeny, there's lots of other flycatchers across the Indian Ocean. And one of the things we'd like to do is to use the lessons that we've learned with the Seychelles paradise flycatcher in terms of the methods for doing a translocation and a reintroduction, to actually apply it to other taxonomic units within these Terpsiphone flycatchers. And on Mauritius, which you'll remember from the phylogeny, there's a subspecies on Mauritius of the mascarene paradise flycatcher, which is there's between 100 and 200 pairs, compared to the subspecies on Reunion, which is very common. And so what we'd like to do in the future is to try and use some of the skills that have been learned on the Seychelles to actually assist with reintroductions of mascarene flycatchers in the Seychelles, in Mauritius. And at the moment, the Mauritius Wildlife Foundation on Mauritius is considering a translocation of flycatchers, of the mascarene flycatcher in the Seychelles from the west of the island on Mauritius across to the east of east of Mauritius, where they're doing lots of reintroductions of other bird species. So what we hope to do is to try and sort of assist some of that work in the future. These are some of the publications that have come out of the work, and many thanks to all of the contributing organisations that have helped with it. And finally, thank you very much.
Thank you very much indeed, Jim. That's a fantastic example. I don't think of how to save or help to save a species.